On behalf of the UCI Susan and Henry Samueli College of Health Sciences and UCI Health, it gives me great pleasure to welcome you to this community lecture series sponsored by the Sue and Bill Gross Stem Cell Research Center. The center is a highlight of the college. The college is now just forming, and it has a school of medicine and a young school of nursing, an emerging school of pharmacy and pharmaceutical sciences, an emerging school of population and public health, and then, of course, the Stem Cell Center, the Cancer Center, and the Institute for Clinical Translational Sciences. This is an extraordinary enterprise. And as I say, the Stem Cell Center is a highlight. It is one of these great examples of interdisciplinary science, the way that UCI makes sing. It brings together faculty from the schools, the 4 schools I mentioned in the college, but also biology and law and computer science. All of these things together because it's at the I'm having trouble with that word. Let's call it the edges of disciplines that incredible discovery takes place because we bring knowledge from different disciplines together, and that's what happens in the Stem Cell Center. There are 50 faculty members engaged in world-class studies, from the basic biology of stem cells to innovative clinical trials. And they're tackling diseases that span the spectrum from ALS and burn and wound healing to diabetes, stroke, glioblastoma, and other cancers. You may have heard last October in the news about a promising clinical trial for glioblastoma. Glioblastoma is an aggressive, thus far incurable form of brain cancer. And Dr. Daniela Boda has a trial, and her patient in this trial was receiving a drug called I'm going to have trouble with words, and I apologize. It sounds like marzipan, but it's morzibim. Daniela will help me. Thank you. And what it does is it targets the cells in the tumor that grow, the stem cells, that have previously been resistant to classical chemotherapy and radiotherapy. The patient that Daniela enrolled was 30 years old, and at the time was given 16 months to live. 2 years later, the tumor is too small to see in many of our measurements. This is extraordinary. The Stem Cell Center receives funding to achieve this type of success from a variety of sources. That includes the California Institute for Regenerative Medicine, CIRM, the NIH, and private philanthropy. In CIRM's 15 years of existence, the UCI has received $125 million of support. This is for infrastructure, training programs, basic research, and regenerative medicine trials. And you may know that CIRM is going to come up on a ballot measure in the months ahead for renewal. I am not a politician, but I'm going to ask you to remember that the benefits of such an investment by the state will allow for successful continuation of training programs that supports graduate students and scientists, that allows for this type of research, and has allowed hundreds of scientists to come here to take short courses to take what we know here and advance it at other institutions. So now it gives me great pleasure to introduce and welcome to the podium the director of our Stem Cell Research Center, Professor Eileen Anderson, who obtained her PhD in biology and neuroscience here at UCI, postdoctoral positions at UCI and Harvard, and became a faculty member here in 2001. Dr. Anderson's research is focused on 2 primary goals. investigating the interaction of transplanted stem cells within the injury milieu and elucidating the role of the inflammatory mechanism in degeneration and regeneration in central nervous system injury. Much of her research bridges the boundary between the basic and translational efforts to ameliorate the problem and to cause regeneration and recovery of function. She was appointed the director of the Stem Cell Research Center in 2017, and over the past 2 years, she's led us to incredible strength on strength from where we've come. So thank you for being here, and thank you for letting me welcome Eileen to the stage.
So I just want to say a few brief words. First is thank you all for coming tonight. This really means a lot to us as a center. I was saying a few words earlier, just a few minutes ago. About a year and a half ago, in 2018, we launched an effort to do outreach to the community and really expand what those outreach activities have been. We've hosted 15 community lectures, not counting these evening lectures that are linked to our annual symposia. In those 15 lectures, I'm just wildly thrilled to say that we've had over 2,200 members of the community come to see them. So give yourselves a round of applause for being here tonight. We really, really appreciate it, and we're glad to see you. So just one quick word. Those lectures allow us to tell the community about the things we're excited about, our research programs here, which highlight both the basic science and the clinical science that's being done in our Alpha Stem Cell Clinic. But more broadly, they give us a chance to talk about where we see our vision, the future of stem cell research and regenerative medicine, not just at UCI, which we think is incredibly important, but on a state level for all of California. And so again, we're thrilled to have you be here tonight to listen to that. One quick word in that. So Dr. Goldstein mentions funding and the diversity of funding in terms of our resources in the center. And we have a number of donors in the audience tonight, and I really want to give a special shout out there because of course that federal and state funding is critical to the research that's done here, but it all gets started somewhere. Those grants are tough to get. So when Dr. Golub was director, he launched a seed grant program. We have 5 years of data on that seed grant program now. With about $450,000 of donor-funded seed grants, we've actually garnered almost $12 million of federal and state funding. So that's a 25-fold return on investment, and it's due to people like you that are sitting in the audience. So again, thank you very much for your time and for your dedication, for your interest in the center. With that, I just want to introduce our speaker tonight who we're really delighted to have here. Dr. Sally Temple is a friend, but also really an icon in the field in terms of neural stem cell research. She's a co-founder, I should say, for the Neural Stem Cell Institute in New York. She is a past president of the International Society of Stem Cell Research, which is really the preeminent organization for stem cell research internationally. She is a MacArthur Fellow. She has founded 2 companies. She's extremely active in not only stem cell research and translational research, but also in ethics and policy and things that are very important to us and that we talk about in our ongoing lecture series for the community at the center. So with that, please join me in welcoming Sally to the stage.
Thank you. Thank you. Thank you so much. Thank you for that very generous, kind introduction. And thanks everyone for coming out tonight to learn about stem cells. I feel this is a very special community. I've really enjoyed my visit. It was a fabulous symposium today. And thank you for staying later tonight and listening to my talk. So I need a new brain. I often feel like this. I need a new brain, Where should we go? And so my goals in this talk are really to tell you about the very exciting things that are happening in stem cell research for brain repair, for brain, retinal, and spinal cord repair. I also want to get over some very important messages, that there are different types of stem cells. You have to really think carefully about that. And that it is very important to be an educated consumer of stem cell products. And you will understand why I underscore that a little bit later on. So first of all, just so I can see if we're all on the same page, I wonder if I could have a show of hands for anyone that is getting older. Yes? And if, thank you. And if you didn't put your hand up, could we talk after this? Yes, so aging. We are all succumbing, and we have different perspectives depending on where we are. So I found this. I love this drawing here. This is the mountain of life, starting as a baby and getting older. You peak here between age 9 and 29, and then you see this very rapid decline. So by age 50, so even Dr. Anderson, you will be headed down here, like working with the game. And we see this on the outside, but unfortunately it's happening on the inside too. And it's happening to our central nervous system. That's the brain, the retina, and the spinal cord. You can think of it as the command center, really. And it is the system that allows us to interact with our environment. It's how we see, it's how we have the pictures in the brain, it's how we hear, it's how we respond appropriately. Really, it's how we communicate, move, everything. And so when it's impaired, it has a devastating effect on our lives. And we see this in the number of diseases that affect the central nervous system, and that unfortunately increase in prevalence and incidence with aging. So Alzheimer's disease, of course, we all understand this disease of memory and dementia, 5.3 million Parkinson's, 500,000 stroke, 7 million age-related macular degeneration, 10 million people, and that's in the US. So you think globally, these are really, really feared diseases of aging. And all of them have something in common. They all involve cell loss. So it's not just that things are going a little screwy in there. The cells are actually dying. And so how do we think about replacing those cells and preventing that cell loss and revitalizing so that we maintain quality of life for as long as we possibly can? And this is really where stem cells come in, because stem cells do make new cells. And that is the opportunity that we have now to think about how we might replace and repair the central nervous system. So there's a challenge, though, because every bit of the central nervous system has a unique set of cells. And you can see here in these gorgeous pictures, they're not generated from me, but from other labs. So here's the retina with the gorgeous photoreceptors. Those are the cells that respond to light. Here in the cerebellum, these huge Purkinje neurons with this is like art, right? This is in our heads. Here's the cerebral cortex, the sort of thinking part of the brain, all of these beautiful neurons. These are the excitable cells. The neurons are the electricity-carrying cells that spark and are responsible for our thought and movement and imagination. And these are the cells that are dying. In addition to neurons, we have these diverse types of glial cells, support cells, that help to support the neural function, astrocytes and oligodendrocytes. All of these different cell types can be affected in disease. And so there's a big challenge with stem cell research, which is we have to be able to make these cells authentically. And it's a lot easier to say than done, but there's efforts all around the world to try to make these different tissue types, these different parts of the brain. And a lot of progress has been made, and I will describe some of that. So what might a brain cell therapy look like? All right, so you start off with your stem cells in the lab, and then you have to generate the neurons and glial cells of the specific types that are missing. And that should be done in a GMP manufacturing facility that's to a very stringent level because you're creating a product that has to be put into people. And then that would have to be delivered, surgically delivered. And each of these steps is challenging, but we have made great progress. And here at UC Irvine, I know that infrastructure is in place to do the research. I think you're now building a GMP facility that would enable the cells to be produced. And of course, you have the clinical setting to deliver. So here, you're very fortunate to have this opportunity. And this is really going to be a revolution in the way that we treat some of these devastating diseases of the nervous system. So I want to start at the beginning. I know that many of you have been here before and you know what a stem cell is, but I figured I would start with the basics because it's really important to understand that the stem cell these are the definitions. So a stem cell, I think this is running out, so I'm going to use the large pointer. Oops, yeah. So a stem cell can self-renew. It's a regenerating product. It means it can make more of itself. And in addition, it can differentiate into one or more cell types. And that's it. That's the two features that you have to remember. But there are different types of stem cells. So early on in the developing embryo, when it's at the blastocyst stage, there's a tiny collection of cells here that consists of stem cells, and you can grow them into an embryonic stem cell line. So this is at the embryonic stage. There are stem cells through fetal development that are building the organs in the body. And then there are stem cells active right through life. And even in adulthood, there are pockets of stem cells that you're familiar with, blood stem cells, skin stem cells, and so forth. So if you look at these different classes then, these are tissue-derived stem cells, and the embryonic ones are pluripotent stem cells. And these can give any of these tissues. So stem cells come in different flavors, and their properties differ also. So if you look at these embryonic stem cells, I mentioned to you they're pluripotent, which means that they can make any cell type in the body. There's a related cell, which I'm going to describe in a few minutes, but it's called an induced pluripotent stem cell. It also is pluripotent, so it can make any cell type in the body. And both of these cell types have unlimited ability to divide and self-renew. So you think about that. You can have one line that's pluripotent, it can make any cell type in the body, and it can renew forever. So that is an enormous resource that we have to make new cells. And these are from human cells, so you're making human cell products that can be used. But this unlimited proliferation, there's a double-edged sword. because you have to be careful about tumor formation. If anything can divide and divide and divide and grow, you have to be careful about this. So this is why we must be very careful about how those products are manufactured and how those products are tested. And that's the sort of rigorous research that you have to do before you enter a clinical trial. And it should be done under proper oversight and done with the FDA oversight as well. So these are pluripotent cells, and now you've got those adult or tissue-derived stem cells that I also mentioned. And these are different because they are biased to making the cells of the tissue of origin. So if you have a stem cell in the skin, and you know that when you cut yourself and it heals, that's because there are stem cells that are activated to repair. So they make skin. And, you know, bone marrow transplant that's got blood stem cells in it, blood stem cells that make new blood every day. And in a bone marrow transplant, you're given those stem cells as a therapeutic strategy, or a skin graft for a burn victim. So adult cells are biased towards making particular cell types. They've been used for many years. They're a known quantity, and they're safe. And they have less ability to divide, but there's some stability and safety that comes with that as well. And so when you think about stem cells, I can always tell people say to me, oh, I had the stem cell. I think, do you really know what happened when you had that procedure? Because if you think there's just one stem cell, then you're wrong, because this is truth. There's a lot of different types. Okay. So these pluripotent stem cells have been discovered really since 1998, not that long ago. And the fact that we can use them to make any cell type in the body is just really an extraordinary opportunity. So the embryonic stem cells that you're familiar with that come from the blastocyst is one set of cells. But there's another way to make pluripotent stem cells, and that is to take some tissue, maybe a blood sample or a skin sample, put that into the lab, and then add 4 genes, these very specific factors that can be added into the cells, and those cells now become changed into a pluripotent state. You know, at the time when this was being discovered around 2006, 2007, I think we were all skeptical. You know, I couldn't possibly do this. It's like turning things back in time. But, you know, it really does work. And it works so well that just a few years later, Shinya Yamanaka got the Nobel Prize for this discovery. And because I come from Albany, New York, I like to point out that the year before, he got the Albany Prize, which I'm sure you've all heard of here. Yeah, so I'm sure the Nobel Committee just, you know, heard what we did and realized it was a really good choice. But he revolutionized what we can do in the lab and what we can do in terms of making cells for brain repair. So what's at the leading edge of what we can do now? And just, you know, bear in mind, this is still really recent, and here we are already thinking about targeting diseases like Parkinson's disease. You probably know some people who are suffering from Parkinson's, where you have inability to move or to initiate movement, tremor. It's very debilitating. And this is due to loss of cells in a very small part of the brain called the substantia nigra. So here it is, and then these cells actually have beautiful long processes that go out and innervate other parts of the brain that are important for movement control. So here's a picture of an image of a brain. Isn't this just the most beautiful structure, right? Isn't it gorgeous? And you see all the different parts. Now you understand why the cells here look different from these other regions. And here is the place where the dopaminergic cells that are lost in Parkinson's disease. So very precise loss, and yet you have this very devastating effect. And so what can we do to replace those cells? So I don't work on this disease, but my friend Lorenz Studer, who is a pioneer in this area, was very kind and gave me some slides so that I could share with you the sorts of lab experiments that he did in order to advance this cell therapy. So first of all, he had to take those stem cells and make them into the dopaminergic neurons in the laboratory. And then, having done that, and here's a picture of, this is in this case a mouse brain, he then had to inject those cells. And he's used a fluorescent dye so that we can locate the cells. You'll see that they did an injection and they're able to see the cells, and they were differentiated into the dopaminergic cells that are marked by tyrosine hydroxylase here and FOXA2, two genes that are critical to show that they have the right cell types. And then the animals that receive that then do some tests. So they put them into animals that are a model of Parkinson's disease. So the animals themselves struggle with stepping, for example. So here's the stepping of an animal that has not been treated here in the red bar. And then when they have been treated, you see this increase in the stepping ability. So clearly this animal is moving better because it's had the graft. This is a test also in which the mouse or rat will be put inside a cylinder and will reach up, and it demonstrates that there's a significant benefit of these particular cells. Now, they also had to do some experiments in rhesus monkey. This was required in order to get the approval from the FDA to, or at least the allowance to move into the clinic. And again, they injected the cells and stained them for these two key genes and demonstrated, as you can see with the green and red staining, that they had those two markers. So that was a success because it showed that the cells could be transplanted, survive, and have benefit in vivo. And just to show you some of these gorgeous neurons as they are differentiated, so you can see all the fine processes that they make. So I just wanted to show this movie. So here, if you look at this, this is an animal that has been treated, and at the 2-month time point, you can see it can't move this paw. But a couple of months later, at the 4-month time point, now it's beginning to be able to move it. So there's 2 months, and then keep watching. You'll see now that it's moving the paw. So here's an animal that was injured, that the cells, once they were transplanted in, those cells that are key for Parkinson's disease could help that animal recover. So this is very, very exciting. And he has then gone on to develop this further and has already filed an IND application to the FDA. So I just want to review the timeline for this. So the discovery of embryonic stem cells here in the late 90s. The discovery of induced pluripotent stem cells, right? 2007 for human. Production of dopaminergic neurons from those cells just a few years later. And then GMP manufacturing, animal studies, transplant and IND filing and IND allowance last year. in the US. This is really rapid, right? This is very exciting. And it's not just here in the US, they're doing this in Europe. Lorenz was fortunate to found a company, Blue Rack Therapeutics, which is then able to carry this forward with investment. The Riken Institute, Jun Takahashi is doing this. Here in California, Aspen Neuroscience. And I just want to highlight the fact that all of this work that enabled it, it's really a community effort. Scientists build off of other people's work. And here in California, CIRM invested, I think, over $600 million towards this effort, which is really, really important. So now I want to give another example because this one is very close to my heart. My mother had age-related macular degeneration, and I know what a cost that is to one's life, one's ability to read, recognize faces of loved ones, go out shopping, read the medication, all of these things she was not able to do. And it's really a devastating disease. So with macular degeneration, what happens is you lose central vision. So it's actually a very small piece of the retina, just a couple of millimeters across, that's affected in the eye. But when this happens, it's where all of the work is done. Just imagine if everywhere you look, there was a big black blob and you couldn't see anything, you couldn't look around it. That's what it's like. And if you look with age, it increases so that 1 in 5 people over age 75 have age-related macular degeneration. So we really do need to think of a way to combat this disease. So macular degeneration is due to a loss of this little thin layer of cells that's in the back of the eye. You know, you look in the eye and you see black. That's because there's a pigmented layer of cells in there called the retinal pigment epithelium. And these are central nervous system cells that die in this disease. And when they die, the photoreceptors die. Remember those beautiful light-responsive cells I showed you? And so that's why we begin to lose vision. So there is, again, a lot of effort to replace these retinal pigment epithelial cells. So again, what would this look like? You start with the stem cells, is getting familiar, right? Now you have to make the right cell type. It doesn't look like a dopaminergic neuron anymore. These are these gorgeous hexagonal cells called the retinal pigment epithelial cells. And again, the concept is that these will be delivered in an operating room and injected into that macular region in the back of the eye. And I know that you have research going on here at UC Irvine with Magdalena Sila, who I think is here, yes, already moving this type of program ahead. In that case, I know even doing more of the retinal cells. So if you look at this, where are we in terms of progress? Again, the exciting thing is that we know how to make the cells, and we're already in the clinic for this particular disease. The embryonic stem cells, induced pluripotent stem cells I mentioned. Look at, there's a lot of different companies and institutes that are involved in pushing this forward. And it's very important to recognize that company involvement is important because they help bring in the money and the funding that can extend these therapies and turn them into products. So to see this level of activity is very important. There's also an adult stem cell that comes from the eye itself, And this is what we're doing, so I have to mention it. So we're growing cells from cadaver eyes and injecting them into patients in much the same way that I described. That is the goal. We are just about to file our IND. We have a little scheduling difficulty because we were going to file in May. Does anyone know how long the FDA have to review when you put in your investigational new drug application? Do you know how long? Just how long do you think the FDA has? How long? 2 years. Okay. They have 30 days to review it. Now, our R&D application is over 4,000 pages. Over 4,000 pages. And I just can't imagine the workload that they have, this group of people. So we're going to send that. Now, during that 30-day period, you have to be there on the phone if they call you with any questions. And I said, well, we're going to put it in May. And then my daughter said she was pregnant, and she's due May 15th. So now like, oh, do we go earlier? Do we go later? Oh, it's all good, right? A month is good. But we're very excited about this. And we actually just started a company called Luxa Biotech, and we're excited to move that ahead. So here's more of a challenge. Alzheimer's disease, 37,000 people a month diagnosed. And I was telling someone tonight that my mom had AMD, and then she got diagnosed with Alzheimer's. And she said to me one day, she had a very funny sense of humor, which she didn't really lose. And she said to me, you know, I thought it was really bad when I lost my eyesight, but losing your brain is so much worse. Like, okay, Mom. Yeah, so, and it is, right. And just so that you understand the level of cell loss, you know, here is, now you got used to pictures of brains. Looking down on the brain, you see the cortex here with all of these little, like, granules. This is a healthy brain. Now, in Alzheimer's disease, you can see suddenly, you know, this hole in the brain, the ventricle, which is where the cerebrospinal fluid is so much bigger because all of the tissue is shrinking down here. You know, this is very widespread cell loss. You know, the different types of atrophy. You can see this really is a devastating loss of cells. And so, unlike Parkinson's disease, where you had a definite, you know, number of cells and very defined space that you could target, this is a very different, it's orders of magnitude more complicated to think about how you might replace cells. But I think stem cells can be really helpful because we can use them to discover drugs. And what this requires then is to make those induced pluripotent stem cells I mentioned, not from healthy people, although we need those as controls, but from people who have Alzheimer's disease or related dementias, especially dementias where we know there's a genetic underlying cause. So then we can take a skin sample, we can add those 4 genes, and then end up producing induced pluripotent stem cells, which we differentiate into the cells that are affected in the culture dish. And then we can start doing drug screens to see how can we reverse the phenotypes in these cells. And what we have found is that as we're modeling this, we're finding that indeed the patient's cells are dying more. They're losing the connections, the synapses. They're showing changes that we associate with dementia, with Alzheimer's disease. And so our hope is that when we do these drug screens, that we're going to be able to reveal compounds that will help slow down the disease process. And this is being done in labs across the world. That's the exciting thing. And people are sharing that information. And we're seeing progress in understanding why the disease occurs, what the disease mechanisms are, and what targets we can intervene with and hopefully slow this down or maybe prevent it from happening at all. That would be the dream, right? So one of the models, just to end, I want to show you, is a 3D organoid. I don't know if people saw the news about these organoids that people are growing from stem cells. So we are doing this right now. You take the stem cells, you're familiar with that, and then you can culture them in the Petri dish and create these three-dimensional structures. So imagine that you can actually see them with the naked eye. They're about 1 millimeter across. You can look down and see them in the Petri dish. And here what the cells look like when you section them. And they look like they have brain cells here. And we're using these now to study the process of disease and also to use them to test drugs. This is a quick movie, which I just wanted to show you because when we do microscopy on these brain organoids, we find these cells are very dynamic. It's really remarkable to think that this is going on in our heads, right? This type of cell movement, that's all part and parcel of this amazing central command system that we have. So I know we started a few minutes past, so it's okay. I just wanted to go on then and summarize what I've talked about so far, that there are a lot of promising efforts around the world using stem cells for central nervous system repair, for Parkinson's disease, for macular degeneration, also for several white matter diseases like multiple sclerosis, spinal cord injury, retinitis pigmentosa. We're seeing a lot of these CNS diseases now being approached with a cell transplant or with disease modeling and drug screening for diseases like Alzheimer's. We do, though, have to be cognizant of the cost and the time. There's many kind of almost, it's, how can I say it? It's depressing when you understand how much this costs, right? It costs $1 billion and it takes 10 years to get one drug. I think now that we're getting better at this process. I showed you really that things are speeding up now. But at the same time, we do have to be realistic. These things are not going to happen tomorrow. They take time. They take investment. So we just have to keep, putting one foot in front of the other. And, we see this growth also in the regenerative medicine field, in regenerative medicine companies, 466 companies in North America alone in this regenerative medicine space. But with that, there really is, you have to be very careful. There's the need to develop these therapies responsibly. And if we don't do that, if we ignore the science, start making false claims, there's a lot of greed in there, then you can cause patient harm, which degrades public trust, and it harms the whole field of stem cell research and regenerative medicine. And so we really have to be careful. What's the work that's going on here at UC Irvine, the work that's funded by CIRM, is carefully scrutinized, it's carefully reviewed, it's done in a carefully regulated environment. But not everybody is doing that. Other people are just hanging the shingle up and saying, hey, I have a stem cell for you, and as you know, it cures everything. And then you get really sad stories like this, where patients paid $5,000 to receive so-called stem cell injections into the eye, and they were blinded. They lost sight. So this was a few years ago, this U.S. stem cell clinic was the clinic that they highlighted here. This type of thing, though, is going on everywhere. Now, how did those patients find out about the clinic? They went to a government website, clinicaltrials.gov, because you can list anything here, right? It's based on an honor system. So people are putting their information there. And if you look at clinicaltrials.gov, there's the US Stem Cell Clinic there listed as having a completed study or a withdrawn study. Be cautious. There is a disclaimer at the site that says that they really are not responsible for the information that's posted, but some people think, because it's a government website, that it is, and you just have to be very, very careful. And if you think this was just a couple of days ago, they're listed right there. So I was on a plane going from Albany to Florida once, and this guy sat next to me, and my husband's there, and he said, what do you do? And I said, oh, we're working on a stem cell therapy for macular degeneration. And he said, that is all you are going to talk about for the next 3 hours. Like, oh, he must really be interested in this. So it turned out that he lived in Florida, he was a lawyer, and he was suing the US Stem Cell Clinic. Wow. I mean, what's the chance, really? So here he is, and this is an article that was in the Washington Post, and he showed me videos of this, wonderful woman actually being injected. They had taken the video and injected, showing her having the injection that blinded her. Awful. Not long after that, thanks to his work, thanks to the Washington Post, the FDA filed a permanent injunction against the US Stem Cell Clinic. That is good news, maybe, because 2 days ago, this is their website. There they are. There's Kristen and Camilla. You can schedule an appointment. You can find out all about the stem cell therapies that they're offering. So be very, very careful. So what do? They do liposuction, and they take what they call stem cells, and they inject them into the areas of injury. It can be from fat. It can be from bone marrow, too. They're advertising that now. And they're saying, this is all the things Parkinson's, ALS, stroke, any of those things, they can cure it with this. And people are mortgaging their homes, and, it's just a terrible situation. And it is going on today. And so it sounds great, because it's like, oh, we're going to remove some fat, which you don't want, and then we're going to build some other piece that needs building. But I want you to think about this. I want you to think about what is in the syringe, right? When she was sitting on the bed and I saw the video, and the guy who was not even wearing a white coat and who wasn't a doctor, let alone a surgeon, came to her with that needle, what was in it? Were they your cells? Were they stem cells? Or were they human? Did they really make the cell that you want and for your indication? Had they been tested for HIV? These are the sorts of things that is the reason that we work in a regulated environment. This is from the FDA website, actually, because the FDA was set up to prevent this type of thing years ago. the stem cell snake oil. And you have to learn what the red flags are. If they're charging you money, that's a red flag. If they're claiming that it will cure everything, that's a red flag. And really, I would recommend that you check out this website. It's a closer look at stem cells. This is a website that the International Society for Stem Cell Research puts together, 9 things to know about stem cell treatments. It's in lay language. It's been translated into other languages. It really is, there's a patient handbook. It's really something that's useful. And if you hear of someone that says, oh, I'm thinking of having stem cell, you're going to go check that website out and be an informed consumer. You know, as scientists, we are supposed to be visionary. We're supposed to be forward-looking and optimistic and think about where our discoveries might lead. But we temper that. We temper it because we are critical. We test things out. We have to show by repetition and rigorous research a proof before you go into patients so that patients will trust what we're doing. And this is absolutely the opposite, the hype, the false advertising, the pay to participate. And I just wanted to highlight that CIRM really, by investing in the high quality research and infrastructure that you have here in California, in the knowledge and in the outreach, really helps protect the public as well as advancing the fields. And that's really, you're very, very fortunate to have that here. So are stem cells the answer? Well, I think they're definitely part of the answer, and I do feel that we have a lot of possibilities ahead. I've said, be a critical consumer. And this enterprise really depends on public support. The realization that science makes medicine, that what we're doing here and in the labs here and around the world is helping to generate medicines that are benefiting human health. We have to have a realistic timeline and understand the investment, but I believe we have to start now and we have to keep pushing forward. We can't let the cost and the time delay us any longer. And we have to join forces. You know, Eileen mentioned that I've been fortunate to have some opportunity to talk to people in government, to do some policy work just as really as a lay person, just to go and advocate for science, for stem cell research. Kristen Gillibrand came and visited our facility. It's great to have people from government come by and teach them about what science is about. I have walked the halls and rolled the halls with my very good friends who advocate for stem cell research and spinal cord injury research in New York. I recently gave testimony to Congress in defense of fetal tissue research. Here also we are talking about stem cell research at the Capitol, and the only one that my kids really think is cool is my 7 seconds on the Dr. Oz show, talking about stem cell research. But it's very important, and thank you for being a part of it and for your kind attention tonight. Thank you.
If you could all just join me in thanking Sally again. We have a little artistic sort of plaque for you. And just very briefly, we're going to take questions in just a moment, but I'd like to invite Jonathan Thomas up to the stage because we have a special guest tonight. John is the chairman of the ICOC for CIRM, so the Citizens Oversight Committee, and has been for some time. And I think you might like to have a few words to say, and then we'll open up for questions. I have a few starters, and we'll go from there.
Thank you, Eileen. It's wonderful to be here at UC Irvine. First of all, to be able to follow one of the giants in the field, Sally, is a great honor, and wonderful listening to you and descriptions of all your and other people's work in this most important area of neurodegeneration, which affects so many people. I want to shout out to UC Irvine, Eileen, and all of the incredible work that's being done here in stem cell research. Courtesy of the voters, we have been able to do our modest part. You heard, or may have heard, we've contributed, again, $125 million to UC Irvine over time, and that money has been put to tremendous use across a broad spectrum of different types of diseases and conditions. I did want to add one sort of interesting little anecdote following Sally, which I don't know if she's heard. She talked about this induced pluripotent stem cell discovery by Dr. Yamanaka, who happens to split his time between the University of Kyoto and the Gladstone Institute up in San Francisco. And when he got his Nobel Prize, the fellow who runs the stem cell program, now the head of the Gladstone Institute and current successor to Sally is president or chair? Were you president of the International Society for Stem Cell Research? His name is Deepak Srivastava. Invited people over to the Gladstone to Skype in Shinya, who was in Kyoto, to talk about the award of the Nobel Prize. So he gets him up on the screen, and Deepak says to him, so what was it like getting that call? And he says, as Sally knows him very well, a very modest, very low-key fellow. He says, well, I was on the floor in our kitchen fixing our dishwasher. And the phone rang, and my wife answered and said, "Dear, it's some guy from Sweden." And I got on the phone, and they told me the news, and I thanked him very much, and I went back and continued fixing our dishwasher. Had you heard that story before? Okay, you can picture that. If you know Dr. Yamanaka, you can picture this. Also, by the way, at another speech, just run a marathon, and this lengthy introduction to his speech talks about how he was training, and he was determined to beat 4 hours. And he finally finished the race, and he puts up this slide, and as you know, they have, at the finish lines of these things, they have clocks which have seconds down to the second above the tape above the finish line. And there he is running through, looking in great form, and the clock reads 4.001. It's 1 second over four hours. Anyway, so I could tell lots of stories like this, but briefly on CIRM, as you know, created in 2004 by the voters, Proposition 71 set up the agency to fund stem cell research throughout California, and importantly, authorized the state to issue $3 billion in general obligation bonds to fund the grants. Since 2004, we have given out over 1,000 grants. We have funded over 70 different entities made up of med schools, research institutes, and biotech companies, all in California. We have research going in over 40 currently incurable diseases, from all the big ones, heart disease, diabetes, neurological disorders, vision disorders, et cetera, down to many rare or so-called orphan diseases. Fully 30, I think the last number is 31% of our portfolio is in neurological disease of one sort or another. So it's very important and very challenging, as Sally described. There are lots of issues that make it particularly tough. People will ask, I'm sure Sally gets asked, had the question a million times, so what's the next thing? What's going to get cured next? What's the easiest thing to cure? And you can say, well, I'm not sure about that, but I can tell you what some of the tougher things are, and the neurological diseases, particularly Alzheimer's, for reasons described, is one of those. In any event, we've had our first couple of diseases effectively cured, coming out of Don Cohn's lab at UCLA. One is a form of, you may have heard of bubble baby disease, where a child is born with no functioning immune system. They have, Don figured out a way, if as you heard, your blood, which normally has a full complement of immune entities, B cells, T cells, all this sort of thing. If you're born with that disease, you have none of that. He figured a way to go in and using genetic splicing and the process of putting in the correct sequences of DNA base pairs to correct mutations into the blood cells of affected kids and putting those blood cells back in their bone marrow, and they then crank out normal blood with a fully functioning immune system. That disease, which used to be terminal, children dying very young, there are now over 50 kids who are now in school and they get sick and the immune system works and nobody cares. Really fantastic stuff. That's the promise of regenerative medicine. So we are very privileged to live in a state that believes in public funding for medical research. Nobody's been able to duplicate the CIRM model anywhere in the country for a variety of reasons, one of which is we have this weird thing where you can get measures on the ballot. If you get enough signatures, you can put anything on the state ballot, and if it passes, it becomes law. And that's what happened here. It took 1 million signatures to qualify it in 2004. But it's also something that, as you know, embryonic stem cells have political issues attached to them. In certain parts of the country, that's a bigger problem than here. Here, it's not. California, with the frontier mentality we always have had and the cutting edge nature of everything, went and voted for this and created this agency. And we'll see at this time of incredible acceleration of medical research that at the end of the day, people will look back at this era and say, State of California was a massive player in developing cures for any number of diseases, and that all came because you guys voted for Prop 71 in 2004. It's been my privilege to lead CIRM for 8.5 years now, and we're extremely proud of what we've been able to enable, but more than that, we are so incredibly impressed with the talent of the scientists in this state who are producing these budding therapies and cures that are transforming medicine as we speak. So I want to thank all of you for voting for Prop 71, and just to tell you that your money has been very well spent, and to keep an eye out as news rolls out of all the great stuff that has come about as a result of CIRM and the scientists that we've been able to fund. So UCI, keep up the great work. Eileen does a wonderful job, has an incredible set of colleagues, and we look forward to hearing successes coming out of all those labs. Thank you, everybody, for listening. Good luck.
So I'm sure that we have some questions. I have a couple of starters, and then I'd be glad to pass a microphone, or we can shout out and repeat them. The first one that I have that came through is, which cells are better, neural stem cells or embryonic stem cells?
Neural or embryonic? Oh, that's a very, very good question. It really depends on what you need them for. If you want to make any cell type, then you can use an embryonic stem cell. If you want to make a specific nervous system cell type, then you can use a neural stem cell. So you really have to know the properties of the cells and what they're capable of, and then you pick from there.
Do I have an audience question?
Seeing as stem cell research takes a while to progress, how much time do you believe it will take for doctors to begin to be able to practice the use of stem cell medicine on the general public?
Can you repeat that?
Yes. So I think what you said, could you say the beginning again, actually? I couldn't quite hear the beginning. Oh, microphone. Thank you.
How much more time do you believe it will take for doctors to begin to be able to practice the use of stem cell medicine on the general public?
So we are still in the clinical trial phase, certainly for the nervous system, mostly phase 1 and phase 2. So you still have to finish the whole clinical trial process to get that approved for the central nervous system. I think people here probably will have a better idea of how many years that will take, but would it be optimistic to say 5 years if they're in a phase 2 now, right? 10 years. So, thank you.
Yeah, do you see any, I've seen many patients that are getting
Where are you?
placental over here.
Oh, thank you. Hi.
Placental stem cells. These come from harvesting placentas, and they come in little vials, and they're getting these injected joints, IV, et cetera, for various purposes. Is there any role that you see in undifferentiated stem cells, releasing growth factors? And have you seen any kind of research that shows that they do anything?
So that is not approved, right? So it falls into the category that we've been discussing of people basically using things and charging people for it without the real proper clinical trial that should be done. Remember, I said to you that if you have a tissue, it's really good at making, the stem cells in that tissue are really good at making tissue. So if you have blood, it makes blood. If you have skin, it makes skin. Placenta, I don't know what's in there, but it probably makes placenta. I don't know if you really should be putting placenta in your joints, just as you shouldn't be putting fat in your eye. So when you break it down like that, it sounds ridiculous. And, I think that you have to read very, very carefully and discriminate the hype and these false promises with what is real. And unfortunately, when you go to that US stem cell website or any other, you'll see testimonials. People that say, I had this done and it's great and I can walk and I couldn't walk yesterday. But those are just anecdotes. They're not studies. And so you really have to be very careful. You have to think about, is this really going to work? What's the proof? And, work with your healthcare professional for the guidance.
So do you see any role for undifferentiated stem cell treatments? Or do you need specific every time?
All stem cells are undifferentiated, really, all of them. They just have different properties. So the undifferentiated ones in the skin are biased towards making skin. They're still not a fully differentiated product. So, you can use these different tissues as a starting point, but let's do a proper study to see if they're really beneficial, because we're all paying for it ultimately. So, let's do it right. Thank you.
So I had a question. Please correct me if I'm wrong, by the way. So I read somewhere that like mesenchymal stem cells, they have the ability to like modulate a specific system's response and like potentially prevent like pathological response as well. So if that's the case, and I'm assuming that like autoimmune deficiency and like AMD, as you talked about, are like genetic diseases. So why can't you stop that pre-disease then? Like, why does it only have to be post?
So that is a great question, and we do want to stop it pre-disease. But you also have to think about what are the cell types that you'll put in. So with macular degeneration, you lose the retinal pigment epithelium cells, so you want to replace those with new retinal pigment epithelial cells. Mesenchymal cells come often from bone marrow, for example. Again, you shouldn't be putting that under the retina, in my opinion. Yes. Most people's opinion, right? Mesenchymal cells are very interesting because they have a lot of properties that people are figuring out. Unfortunately, there have been a lot of claims, perhaps exuberant claims, about how well the cells can work. But there's been a lot of trials. A lot of the trials haven't worked out, truthfully. But still, those mesenchymal cells are being pursued. And I think we should have an open mind and do it carefully and scientifically and see what those cells might be useful for.
I have a friend that is blind because he had RP.
Okay.
He wants to know how soon he can get into a clinical trial.
So, it really depends, and I know Magdalena's here and she works on that, and Henrik Claassen, of course, is here as well, because there is some really important work that's being done here for retinitis pigmentosa.
Yeah, we saw that in the lab.
Right. So, this work is ongoing now. And often it depends how much vision has been lost, because there is a point of no return, unfortunately. So the sorts of therapies that are being developed now can help slow disease progression. They can help by putting a gene in that's missing, or putting a cell in that's missing, or using cells to provide trophic factors to stop the retinal cells dying. But ultimately, if you have lost all of that vision, then there's nothing there to save. And then you have to think about how in the future you may be able to make an artificial retina or a full thickness retina to replace, which is what I know Magdalena's working on right now. But that's still very much in the research phases. So I think that there is a lot of hope out there if you have some residual vision for RP. And I think that clinical trial is ongoing now, right? But you could find out more from Dr. Anderson, maybe. Yeah.
So yes, the studies here on Dr. Claassen's research in combination with Jay's side are ongoing. There's going to probably be a new study opening very soon. I will guide that everybody that's interested on our clinical trials to go to the Alpha Stem Cell Clinic website and register and leave their information so we can contact them when things targeted to their disease are coming to horizon.
Cool. That's great.
Hi, so I'm just curious, as someone who has years of experience in this field, what is some piece of advice you would want to give to younger aspiring scientists who want to get involved in stem cell research?
Oh, thank you for that question, Dana. Yeah, and we didn't plant it. I always say that there's nothing like discovery. There's just nothing like seeing something for the first time. But there is actually one thing better, and that is watching somebody else do that. Mentoring somebody and seeing them discover something for the first time. That is truly rewarding. So I would say that this is an absolutely ripe time. It's a wonderful time to get in a field that is expanding. And the fact that you're doing both discovery science and things that are meaningful for society, for health, that is really a wonderful raison d'etre. So I think just to go for it and find a lab where you love the mentor, as I think you did, and just go for it. Keep pursuing your dreams. And yes, you have to write grants at weekend, but it doesn't matter. I love writing grants, okay?
So I'm going to interject one because I think it was on topic for you that was handed in ahead. Do neural stem cells age in the lab, and is that a problem?
Oh, yes. They do age. You know, as you culture cells, they change a little bit. And I don't know if it's exactly the aging process, but certainly we're very much aware of how many passages we keep cells because we know that their properties change. We also want, in some cases, to make the cells age. And so we do experiments with genes like the gene that causes accelerated aging, progeria. That's a genetic disease where people begin to look old when they're very young, like an 8-year-old will look like a 60, 70-year-old. And we can introduce those aging genes deliberately in order to model some of the features of aging, because then we can look in the culture dish, we can understand the processes that are going on and hopefully combat it. And then if I said, who's getting older, no one would put their hands up because we'd have the answer. But it's really useful, actually, to have those mechanisms to study aging in the lab. Yeah.
Sally, I wanted just to add to that question. So when you talked about Lorenz's IPS work on Parkinson's, when he's doing that, how does he get the neurons in the dish to effectively be the age that equivalent that you would be when you start developing Parkinson's, such that when you are testing drugs against that condition, you're actually testing it against age-relevant cells?
Okay. So you're not talking about transplant? Are you talking about transplant or are you talking about modeling disease?
Either way.
Okay. So it's such an important point. So when you are transplanting, you have to think about the best cell to integrate. And what is the stage of cell to integrate successfully? And what he found is that if the cells are very mature, they don't integrate well. That's what we found as well for our retinal pigment epithelium. If they're too young, they don't work. If they're too old, they don't work. There's a Goldilocks in the middle where the cells are able to integrate better and survive better. And they found that also for the dopaminergic cells. So when you're talking about transplant, you have to find that sweet spot where the cells integrate better. Now, if you're wanting to model aging in a dish, actually, Lorenz pioneered the use of progerin, that gene that can accelerate aging in the dish. And there are other methods also, but the idea is then you will get the cells to age and show signs of disease so that you can try to identify compounds that will prevent that. So it's really two different timing issues.
So, Sally, you describe for us iPS cells, which are basically stem cells, if I understand correctly. So why do we need embryonic stem cells or fetal cells anymore? Why don't we just all do iPS work?
Lots of induced pluripotent stem cell lines are useful. There's still useful embryonic stem cell lines that are already being progressed towards the clinic, and we don't necessarily want to go back and redo all of that work. So I think that the ES lines, of course, have value as well. The fetal cells have a lot of value. Actually, in order to know that we are on the right track when we are developing a new stem cell for the brain, let's say we want to make a specific cell type, we have to know that we are on the right track and that we're doing that correctly. And so we need access to fetal tissue in order to understand if that process is going correctly. So that is actually very important. So it's really a sort of gold standard so that we know what we're doing.
Maybe one more question, and then we'll offer our thanks to.
So can stem cells be used to achieve immortality or slow aging or extend lifespans? And is there any research being done in that area?
Oh, great question. Yes, I'm hoping so. You put all the stuff on. Yeah, so I think in terms of extending life, a lot of the diseases that we've been talking about are fatal. So if we can extend life by addressing those. Most importantly, we can extend quality of life. Eileen and I worked together on a project for spinal cord injury, and I really learned early on that you have to ask the patient what they want. Because, when I started working in spinal cord injury, of course you're working on motor function, but actually what they might care about is bladder and bowel control, these things that are very important for their quality of life. If we can help people extend the amount of time that they can read, we can put off macular degeneration. That makes a big difference. So I think that there's a lot of opportunities for improving length of life, but especially quality of life.
So I want to thank Sally and thank all of you for your questions tonight. Thank you for joining us, and have a great evening.